Really?
Thank you, sir.
I kind of like that option, actually.
Try not to.
Is it already going?
Yeah, it's already on.
Oh, great. That's cool.
I can never find their stuff.
I typically need an email.
It's my list.
You ready?
Let's go.
All right. Good morning. My name is Jennifer DeLuca, and today it is my pleasure to introduce Dr. Nancy Turner, a research professor in the Department of Nutrition and Food Science here at Texas A&M University. Dr. Turner graduated with her Bachelor of Science degree in Animal Science, her Master of Science in Animal Nutrition, and her Doctorate in Nutrition, all from Texas A&M. Dr. Turner's research areas include suppression of colon carcinogenesis and inflammatory bowel disease with dietary bioactive compounds. Dr. Turner is also involved in the Space Life Science program here at Texas A&M and is currently conducting NASA-funded research to determine the effects of radiation exposure on colon microbiota, microbial metabolites, and the epigenetic state of colonocytes. Dr. Turner is a member of numerous professional organizations and is currently serving as the American Society of Nutrition's representative to the FASEB Publications and Communications Committee. She is also currently serving on the editorial board for several journals, including Advances in Nutrition and Experimental Biology and Medicine. Dr. Turner has received several awards, including the Mentoring Award from the Department of Nutrition and Food Science. Dr. Turner has also co-authored numerous articles published in various scientific journals, including Carcinogenesis, the Journal of Nutrition, and Cancer Prevention and Research. Today, Dr. Turner will present a seminar titled Interplay Between Diet and Colon Microbiota Impacts on Colon Health. Please join me in welcoming Dr. Nancy Turner.
Thank you very much for that wonderful introduction and the opportunity to actually be here and share with you some of the work that we've been doing that explores the interaction between the diet, its impacts on the microbiota, and the resulting impact on our health, because we're really interested in our colon health. For those of you that may not know, colon disease in the U.S. is a growing problem from some perspectives. Right now, colon cancer cases are actually declining when you talk about the general population. But that's just incidence rates. Unfortunately, there's a very high risk of death from somebody who does develop colon cancer. So as a result, it winds up being the second leading cause of death from any form of cancer, second only to lung cancer. The part of the problem that is growing, though, is unfortunately the number of people that suffer from inflammatory bowel disease. The rate of that disease is increasing in the United States. The unfortunate aspect about that and how it relates to colon cancer is the fact that people who suffer from chronic recurrent bouts of inflammatory bowel disease are at a greatly elevated risk of developing colon cancer. Now, a study that was published back in 2009 by Harris et al. Looked at the impact of our aging population on a variety of different types of cancers and the risk of developing those cancers. They found that for colon cancer itself, that the anticipated increase in the rates would be 52% by the year 2030, and that's not too far away. Now, the unfortunate part about this analysis is that it did just looking at the fact of an aging population. It did not consider the fact that we are experiencing an absolute epidemic of obesity in the United States. And unfortunately, there's a very strong promoting effect of colon cancer risk in the obese populations. So when you tie all of these things together, the increasing inflammatory bowel disease, our aging population, and the fact that we're suffering with huge numbers of people who are obese, we anticipate that there are in fact going to be a recurrence or an increase in the risk of developing colon cancer in our country. To give you some context, all of our chronic diseases now take up approximately 75% of all of our national health care expenditures. So that's a huge number. We're actually, drowning underneath healthcare costs in the United States, right? So this is a huge problem. So back when the federal government put together the Healthy People 2020 report, one of the national goals stated in that report is the idea of avoiding preventable diseases from occurring in the first place. That suggests that there is ability to prevent these diseases. And we know, at least with colon cancer, that more than probably 2/3 of the cases of colon cancer are attributable to undesirable diets alone. So that would suggest that there's an opportunity for prevention of this disease. However, to achieve this, really preventing the disease in the first place, we're going to have to do a lot of work. And that's going to wind up being a combination of both diet and behavior modifications in our population in order to prevent the disease from occurring in the first place. So, that's a little context and background on the disease itself. What about the colon? For those of you that may not know, the colon luminal environment is a very complex milieu of molecules. The largest portion of those molecules that reach the colon are the complex carbohydrates, either the dietary fiber itself or resistant starches that are formed whenever we process our foods. They make up the largest fraction of the material that gets to the colon. However, when you start talking about the plant materials that make up those complex carbohydrates, you're talking about cell walls of plants. And if you think about it, a lot of what is contained in a plant is contained within inside those cell walls. And we do not have the enzymes to be able to break those cell walls down. Therefore, there are a lot of other non-absorbed molecules that are carried along with that dietary fiber source, okay, that reach the colon. Therefore, the potential metabolic profile of the luminal contents is contingent on several things. First being the bacterial populations that reside there. There's a large number of bacteria. If you compare somebody from the left side of the room and somebody from the right side of the room, you'll find the potential for greatly differing populations, okay? The distributions of the different bacteria are different among people, and they vary by diet. Okay? And the reason for that is bacteria are substrate-specific metabolizers, fermenters, okay? And so if you provide them with different kinds of substrates, you're going to have the propensity for different types of populations of bacteria to reside there, okay? Okay. Bacterial fermentation, which is their primary metabolic process, winds up resulting in a variety of co-products, in addition to the energy that it generates for them. And some of those byproducts of their fermentation include the short-chain fatty acids. Now, the one that we are particularly interested in the colon health world is butyrate, which is a 4-carbon short-chain fatty acid. And the reason that it is so interesting to us is the fact that it's the primary metabolic substrate for epithelia, the colonocytes lining the epithelium of the colon. Okay, unlike most cells of the body that will preferentially use glucose or maybe even in times of need amino acids as their energy substrates, the colonocytes preferentially use butyrate as that substrate. Well, in addition to this unique characteristic of butyrate use in the colon, we also have the fact that butyrate is an epigenetic regulator of gene expression. This is something that was documented many years ago using cell lines. And what they demonstrated is that butyrate is an effective inhibitor of the histone deacetylases. Now, the outcome of that process is that on specific sites within the histones, you will have an opening up of the architecture of the chromatin, which allows all the transcription factors and other regulators of gene transcription to access the promoter regions of certain genes, leading to the expression of some genes. But not all genes. Again, this is not a ubiquitous kind of response. It is site-specific. And so as I mentioned, that histone deacetylase function was originally detected and studied in cell culture systems. A little while back now, we studied this in vivo to document that, in fact, it does occur when the animals are consuming relevant diets. And so in the study, we had rats that were consuming a standard AIN diet, which is our control diet, that contained cellulose as a dietary fiber source. The reason that we use cellulose is it's a less fermentable fiber, therefore there's limiting amounts of butyrate produced in the colon when the animals are consuming that fiber source. Now, in comparison, we had animals consuming exactly the same diet. However, we included small pellets in these diets, and these pellets were designed to only release the butyrate they contained once they reached the colon. So we effectively wound up having a comparison of a relatively low butyrate colonic environment to a high butyrate colonic environment. And what we found and demonstrated here in vivo was that you would find an increase in the acetylation state, and we were measuring here specifically the H4 histone. Okay? And so this was a significant increase in that acetylation state, suggesting that butyrate was in vivo inhibiting the histone deacetylase reactions. Now, the positive outcome of this change in the acetylation state was a corresponding increase in the expression of P21. P21 is a cell cycle regulator, and these effects corresponded with changes in proliferation in these animals. So we had a net effect of an increase in the expression of this protein that controls cell cycle, and that wound up dampening the proliferation in these animals. We subsequently have studied this in more detail in cell culture systems. Here, these next few slides are going to be data derived from the HCT116 colon cancer cell line, where we incubated the cells either in a standard control media, a media containing butyrate, or media containing butyrate plus the N3-PUFA docosahexaenoic acid. And what we found here was just like what we found in vivo, that there was an increase in the acetylation state of the histones. Here we found a significant increase for the H3 histone and a numerical but not significant increase in the H4 histone. But again, recapitulating the fact that in vivo and in vitro, we have this increase in acetylation of the histones. Importantly, we demonstrate that there's a similar response whether the cells are exposed to butyrate alone or butyrate along with the DHA. Now, subsequent to this, we wanted to look at other epigenetic regulators of gene transcription. So we also monitored the methylation state of the DNA. There is one model of colon cancer that indicates that there's a change in the global methylation state of the genome, as well as changes in the methylation of specific promoters of genes that are involved in regulating various processes, such as apoptosis that occurs in this disease process. And so we were interested in how, or if, at this point in time, because it wasn't discovered, whether butyrate and the combination of butyrate plus the DHA had any effect on the methylation state. And really, to our surprise, we discovered that butyrate by itself produced a reduction in the global methylation state of the mucosa that was similar to our positive control, 5-AZA-DC, which is a known and regularly used positive control for demethylating the genome. However, with the butyrate combined with DHA, although there was a small reduction in the global methylation state, it was not significant relative to the control values. Now, all of this leads to the point of, again, regulation of gene transcription. And in this particular experiment, we were specifically interested in regulating the gene transcription of those products that are involved in the regulation of apoptosis. Over the years, we've demonstrated over and over again that apoptosis is a key regulatory element in controlling the development of colon cancer. So here we were exploring the expression of genes involved in regulating apoptosis. Here I'm showing you the data for just two of the genes that are on the array that we ran, and found that there was, with butyrate exposure, a very large increase in the transcript level of both of these two genes in this colon cancer cell line. Whenever we provided the cells with butyrate plus DHA, there was still a significant increase relative to the control, but it was not as extensive as occurred with the butyrate by itself. And this mirrors our DNA methylation data, where it was not as radical of a change when the butyrate and DHA were used together, suggesting that it's the butyrate itself that, through maybe a combination of effect of the histone acetylation state as well as the DNA methylation status, that's regulating the expression of these genes. Well, that's some great regulatory kind of work suggesting how some of the products of microbial metabolism might be involved in controlling colon cancer. There were a lot of experimental data that over the years suggested this improvement in colon cancer risk when individuals had a high fiber diet. In fact, the first theory about the role of diet in colon cancer was developed by Burkitt, and his theory was that diets that were high in dietary fiber would protect against colon cancer. Well, with all the experimental evidence, there was enough information then to support going into human clinical populations. And there were several intervention studies done. They all chose to use patients that already had polyps, and so the measurement of success was suppression of polyp recurrence on individuals consuming a high fiber diet. Not a single study found that when you looked at the whole population, if you elevate the dietary fiber, that you would suppress colon cancer in these subjects, with, again, the readout being recurrence of these polyps. And so that left a lot of us working in this field scratching our heads going, well, why not? All of the experimental work, the epidemiology work said that this would happen, and now that we get into clinical population, it wasn't observed. Well, some of the reasons that may go into this particular observation is the fact that in these studies, they focused on routinely consumed fiber sources in the populations that were being studied. Now, if you've got a clinical population of people and you're going to be watching them for years at a time, you want to employ a intervention that's going to be acceptable to them and that they're going to be compliant with. So you typically use something that they're used to eating, right? Well, some of the things that kind of rattled around inside my head when I was thinking about this is, well, maybe the fiber sources that we're using just aren't the right ones. Maybe there's something else that come along with those fiber sources that's needed to derive the benefit. They have to be combined with dietary fiber in order to have the benefit and reduce colon cancer occurrence. So we started studying a variety of different foods as well as isolated compounds from specific types of foods to find out if they would be able to help prevent colon cancer. And most of the models that we use are this early stage of the disease in which we find these aberrant crypt lesions that we think are predictive of eventual tumor formation. Now, for those of you that aren't used to looking at the inside of a colon, we've just taken a rat colon, opened it up, laid it out flat. All of these small little white dots with the green rings around them, those are the openings to the crypts that line the inside of the colon. These are all normal, small crypts. Here we have an aberrant crypt. It's enlarged. It's about 2 to 4 times the size of the rest of those normal crypts. But the most distinct thing on this picture is this large cluster of very large individual crypts that are in a single foci. And we think these high multiplicity aberrant crypt foci are the most predictive biomarkers of the disease. So that's what we're using as a readout for most of our studies. So in this first study, we were interested in grapefruit, and I'll be honest with you, we went with ruby red grapefruit because it's agriculturally important to Texas, and Texas was willing to fund the work. So that's why we started there. But ruby red grapefruit have a variety of compounds in them that may have some beneficial effects and had been shown so, at least in cell culture studies. So we went forward with that. And in this first study, we looked at the food itself. So we took grapefruit, we freeze-dried it, ground it up, turned it into a powder that we could incorporate into our diet. And so that's the GFPP and the IGFPP. And then we also used these isolated compounds, naringin and limonene, in our studies. And the naringin was included in the diet at the same level as was found in the grapefruit pulp powder diet. And then the limonene was included at the same level as we had the naringin in. And we found that all 4 of our experimental diets were successful at reducing the numbers of these high multiplicity aberrant crypts. Okay? So all of them were effective. Something about them was capable of producing this protective effect. Now, like any other scientist, I challenge my students, like, okay, so what could be explaining this? And one of the things that we focused on was the issue of inflammation, because remember I told you that people who have inflammatory bowel disease are at greater risk of developing colon cancer. So a lot of our studies analyze things that are regulating or at least mediators of the inflammatory process. So in this study, we looked at both inducible nitric oxide synthase as well as COX-2. The reason we focused on those, if you go to human subjects who have colon cancer, you take out the cancer and you look at the expression of these two inflammatory agents and compare that to their normal non-involved mucosa, you found that there's a greatly increased level of those two proteins in the tumors. Okay, so they are unique to that tumor environment. And what we found is that whenever the animals were consuming the grapefruit pulp powder or the limonene, that we had a significant reduction in the expression of those two proteins. Now, the one thing I think we'll all agree upon, and even the funders of our study would have to agree upon, is the fact that we're not all going to eat a whole lot of ruby red grapefruit every day of our lives. And so getting the exposure to any of these compounds, either the diet itself, the food itself, or the isolated compounds, is probably not going to occur. And so our next set of studies wanted to take the approach of looking at a molecule that we find in most of the plant-based foods that we consume, with the idea being that if we're eating enough plant foods in our diet, our fruits, our vegetables, our grains, our wines, that will get exposed to enough mass of this molecule that it would be meaningful on a daily basis. And so we turned our attention to quercetin. Here again, the quercetin was included in the diet in this experiment that would be just slightly greater than what you find in a Mediterranean diet, so in theory it is an achievable level. It's not something outrageous. And we found that we were able to significantly reduce the number of these high multiplicity lesions. We explored some of the underlying physiological responses that may be occurring that contributes to this, and we found that animals consuming the quercetin diet had lower rates of proliferation in the colon, so fewer cells were replicating in those crypts. And interestingly, we got a significant increase in the rate of apoptosis, or programmed cell death, but only in the animals consuming the quercetin diet if they had been injected with our carcinogen AOM. And so this is a very unique response that occurred only in the animals that are induced to have the disease. And so here we have a very desirable response of hopefully getting rid of those transformed cells because we're inducing apoptosis in those animals. We came back to look at some of our inflammatory mediators. Here we looked at both COX-1, which is the constitutive form of the cyclooxygenase enzyme, as well as the inducible form that tends to be elevated in colon tumors, and found again whether the animals were consuming either injected with saline or the carcinogen, as long as they were consuming the quercetin diet, that we were able to significantly reduce the expression of those two inflammatory agents. Okay, so we did this preliminary work with a couple of different molecules, a couple of different food sources, and found that we had some protection. Well, I still kept coming back to Burkitt's theory. It's like, well, why were we not successful in the US with these clinical trials? And started thinking about the fact that his observations were made in Africa. He was studying the very rural populations. Their diets contained a lot of very small grains. Their diets were not processed, and so they were consuming whole foods. And he was comparing those to people who were in the same geographical environment, of the same genetic background, but living in the cities and eating mostly processed foods in a very different kind of diet, low fiber diet. That's when he said, okay, it must be the fiber that's doing this. Well, the small grains that these people were eating that were protected were sorghums and millets, and in the US, we simply don't eat a lot of those. Okay, it's not a big portion of our diet. And most of the ones that they were consuming are of the ancient varieties that are very high in polyphenolic compounds, very high. I mean, these compounds have, if you measure them in a test tube, antioxidant activities that just blow blueberries out of the water, kind of thing. Very high antioxidant activities. In addition to that, these polyphenols, when you cook these grains, will bind to some of the starch and create resistant starch. Okay, so you not only have the less fermentable fiber of cellulose that you typically find in the Brans from grains, but you now also have a readily fermentable fiber-like material that's getting to the colon as well, along with the polyphenols. So we started studying sorghum grains. Again, using our early indicator of the disease, Aberrant Crypts, where we're comparing acetylose again as our base diet, because if you look at the fermentability of the fiber of sorghum bran, it more closely replicates that you find with cellulose. And we compared the brands then of white sorghum, black sorghum, and brown sorghum. Now, all 4 of these diets were balanced for all the macronutrient content. They had the same level of lipid, protein, and carbohydrate, as well as dietary fiber. So really the only differences between them then would be the unique compounds that come along with the dietary fiber contained in these compounds. And we found that with the black sorghum and the brown sorghum, there was a significant reduction in the number of these lesions. Okay? So we wanted to, again, explore this a little bit further. Remember I told you that these compounds have very high antioxidant activity. When we regress the number of aberrant crypts formed in these animals relative to the antioxidant activity as measured by the ABTS activity of the diet, we found an incredibly strong relationship. You don't normally find this in a lot of physiological systems, so we were quite surprised. I don't want you to walk out of here thinking that it's the antioxidant activity of those molecules that contributed to the reduction in aberrant crypts. But I think what we would conclude from this data is that whatever is in the black and the brown sorghum brands that is contributing to the antioxidant activity is in fact what is contributing to the reduction in aberrant crypt number. That relationship is just too strong not to try to draw that conclusion. In this project, we did what I call the poor man's assessment of the microbial populations, which is to measure short-chain fatty acids in the feces. It's kind of a functional readout of those bacteria. And what we found is that animals consuming the white sorghum had a significant elevation in the concentration of total short-chain fatty acids, as well as butyrate, that important fatty acid I was telling you about earlier. Whereas if the animals were consuming a black sorghum or a brown sorghum bran diet, there was a significant reduction in total short-chain fatty acids. And with the brown sorghum, there was a significant reduction in the concentration of butyrate in the feces. Now this threw us for quite a loop, because that was not what I was expecting to see. However, I will tell you that we've done several experiments with the sorghum brands since this, and we've replicated over and over and over again. So it is not a spurious observation. The thing to think about is that whenever we're measuring the concentration of a specific fatty acid or total fatty acids in the feces, that's the net result of 3 separate possible things that are going on. One is a change in short-chain fatty acid production. The other is the utilization of those short chains by other bacteria that are present there, because they use those as a substrate as well. Or a change in the absorption of those short chains into the epithelium. Okay, and with this experiment, we do not have any kind of data that would allow us to discriminate what may be going on and contributing to those data. From these animals, we also looked at the expression of COX-2 as well as the level of BCL2 in the scrape mucosa. COX-2, again, because it's a pro-inflammatory agent. BCL2, because it inhibits apoptosis induction. And we found that with all 3 of our experimental diets of sorghum brands, that we were able to significantly reduce the expression of those 2 proteins in the scrape mucosa, suggesting an improvement with respect to reduced inflammatory tone, as well as an enhanced susceptibility to apoptosis induction. So, all the data that I've shown you so far point to the fact that some of the bioactive compounds that are contained either in the foods or those isolated compounds that we're using are able to reduce disease occurrence through mechanisms that may involve changes in the inflammatory tone in the colon, or changes to the microbial populations and or their metabolism. Okay? Since I told you that inflammatory bowel disease is a strong promoter of colon cancer, we wanted to find out if we were able to reduce the kinds of problems that develop during an inflammatory bowel disease. And so we went to an inflammatory bowel disease model using DSS as the pro-inflammatory agent. Now again, the development of and the severity of colitis that people experience is in part attributed to a dysbiosis that occurs in those individuals, with dysbiosis being just a simple perturbation. We can't really define what a healthy microbial population is yet. We're getting closer every day, but we know that when you compare healthy versus a diseased individual, there's a very different microbial population, okay? And again, recurrent colitis is a promoter of colon cancer, so that's why we went into this area. And I include this cover because I just simply love the image here of all the bacteria. It was an issue associated with inflammatory bowel disease. So once again, we return to our sorghum varieties. Since we had such a promising effect on colon cancer, we wanted to explore that here as well. In this experiment, though, we used 2 control diets. One being the one we've used before, where it's not a low fermentability fiber cellulose, but the other is a highly fermentable fiber source, pectin. And so with pectin, you're going to get lots of butyrate production. In this experiment, we used the same black sorghum bran and sumac sorghum bran, the brown one from before, as well as a high tannin black. And the high tannin black is a unique variety that was developed here at A&M that includes both the condensed tannins present in sumac as well as the proanthocyanidins contained in the black. And what we found, and this kind of replicates some of what you find in the literature, is that a readily fermentable fiber source leads to an enhancement of colitis symptoms. And what I'm showing you here is the change in injury score from our control animals to those exposed to DSS, as well as a change in NF-kappa B activation in those same animals. And what we find is that the readily fermentable fiber leads to the greatest enhancement of injury and the greatest increase in activation of NF-kappa B, whereas cellulose has a limited effect and is similar, in fact, to the changes that occur with the animals consuming the black sorghum bran or the sumac sorghum bran. Well, what happened to the bacteria? We finally got to the point where I had enough money to do some sequencing work on the microbiota, and these colored graphics shows you the phyla level comparisons among those diets. And I think it's fairly easy for anyone to see that there are major differences based on the diet that the animal was consuming. Okay, you have cellulose with no polyphenols contained in it, black bran where we have the proanthocyanidins, the sumac where we have condensed tannins, the major changes relative to the cellulose, and then high tannin that really is kind of in between the black and the sumac. And so these polyphenols that are unique to each one of these types of sorghum brands seems to be having its own unique impact on the microbiota. Now, when we look at the changes in the phylum level, we find this kind of interesting pattern where any of our sorghum brands led to an increase in the relative proportion of the Bacteriodetes present in these animals. And for most of them, that led to a corresponding reduction in the level of the Firmicutes that were there as well. Now, if we look at these actual numbers and look at the ratio between Firmicutes to Bacteriodetes, we find some very striking differences. And the reason that this ratio is purported to be important is that if you study individuals who have inflammatory bowel disease or individuals who are obese and compare them to their healthy counterparts, you find that those diseased individuals have a very high Firmicutes to Bacteriodetes ratio. If you look at our cellulose animals and look at this ratio and compare it to any of the sorghum brands, you see that it is greatly elevated. Okay, and this is at day 47, and that's after 2 bouts of inflammatory challenges with DSS. Okay, so these animals have been challenged twice now. They're in the recovery phase, and this is what that ratio looks like. We go to day 62 and look at the ratio for the DSS animals and cellulose and compare it to those at day 62. You find that that ratio is getting even worse. Whereas in the animals that are consuming the sorghum brands, the number really doesn't change. In fact, for the most part, if it does change, it goes down. And if you compare the control animals to their DSS counterparts on any of the sorghum brand diets, you find that those numbers are really not different. So, we've started dipping our toe into the obesity world. And the reason that we went in this direction is those last observations, where we found that we had an improvement in that ratio that would be also beneficial for people who are obese, right? We know that obesity creates inflammation. We know that obesity promotes colon carcinogenesis. We realize that perturbations to the microbial populations caused by diet may drive obesity risk in some individuals. So that led us to ask the question then, can these dietary bioactive compounds that we've been using impact the bacteria in overweight subjects in a way that beneficially alters their metabolism? So to do this, we had a set of subjects who were overweight to obese. It was a crossover design. We collected samples at baseline prior to any treatments, and then gave the individuals a breakfast cereal that they were supposed to replace their existing breakfast cereal with. That was the only modification to their diet, their exercise, or anything they were supposed to do. We even told them, please don't try to lose weight. Okay, just change out your breakfast cereal. And so they were consuming either 50 grams of breakfast cereal in the morning or consuming 50 grams in the morning and 50 grams in the evening. It's kind of a snack. Okay. The breakfast cereal, as you can see here, actually looks like Cocoa Puffs. It did taste good as well. I was really pleased whenever it made it easier for the people to be compliant, right? And we made that from our sumac sorghum. Again, that's the sorghum variety that was the most protective against colon cancer and did provide some protection against the inflammatory bowel disease model. And so it included the whole grain as well as some additional bran from the sorghum bran, so we can have higher fiber content and higher bioactive contents. And from these subjects, we collected samples to be able to measure the metabolome in the plasma, look at lipoprotein subfractions, and characterize the intestinal microbiota. So, these are the metabolomics, a sampling of the metabolomics data. There's quite a big collection of it, actually. And panels A, B, and C here are 3 microbially derived metabolites of polyphenols. Okay, these molecules are thought to be only derived from microbial metabolism. Okay, so the intestine would be the site of their derivation. There are pathway maps showing how you can generate all of these molecules from a very small list of polyphenolic compounds. In panels D, E, and F, I'm showing you some examples of gamma-glutamyl amino acids. Okay, we see that the metabolites of the polyphenols are significantly elevated. So on the left, we have the low control time point and the high control time point, and then the low dietary period and high dietary period. So both of them are constructed the same way. We see that we get, again, an increase in these metabolites of the polyphenols in the plasma, whereas we get a strong, significant reduction in the level of the gamma-glutamyl amino acids. Now, the importance of these observations are that there are studies out there demonstrating that molecules like 3-hydroxyhippurate are found at very low levels of individuals who are having the greatest problems with glucose tolerance. Okay? And the elevated levels of gamma-glutamyl transferase activity, which produces these amino acids, are associated with cardiovascular risk as well as impaired glucose tolerance. These data would suggest that in our otherwise overweight and obese subjects, we were able to potentially have some major changes in the metabolic profile of their plasma that could contribute to an improvement in cardiovascular risk and glucose metabolism. To study the cardiovascular risk a little bit deeper, we looked at lipoprotein profiles, and here we're looking at subfractions of LDL and HDL lipoproteins. We did this work up in Dr. Walson's lab, and what we found is that there was, after the individuals had consumed either the high or the low level, that there was a significant increase in the LDL2 and LDL3 subfraction, a decrease in the LDL5 subfraction, and at least for the low dose of the sorghum grain, there was a significant reduction in HDL3B. There are some studies out there characterizing individual cardiovascular risk in subjects where they look at these various different distributions of the LDL and HDL subfractions. These changes would correspond to a more cardioprotective profile in those subjects. So what about the bacteria? Just to give you a couple of examples, we found an increase in Faecalibacterium. This is after the low dose of the cereal or the high dose of the cereal. There was a nearly significant increase in Akkermansia. These are both interesting bacterial groups. Faecalibacterium is associated with a low inflammatory tone status. And actually, there are clinical trials going on now where they are giving diabetic subjects Akkermansia in an attempt to try to improve glucose tolerance. And so we found an improvement in both of those. And we dwelled down a little bit deeper in the Faecalibacterium, we find that at least with a high dose of the sorghum bran, that we get a significant increase in Faecalibacterium prausnitzii. And this particular bacterium you can almost not detect at all in people who have inflammatory bowel disease. It's essentially missing, okay? And you see that in our obese subjects, where basically most of our observations of the control period are bouncing around right around the baseline of this graph. But after they consume the diet, you get this expansion in the observations, and there are very few individuals that have little to no detectable prausnitzii in their fecal samples. So our last study that I want to tell you about is dried plums. This is the most recent study that we've conducted. It's a follow-on because some of the compounds that are in dried plums we've played with before in our lab. So it made a logical extension of the work. Now there's some cell culture work showing that phenolic extracts from plums are protective and that they inhibit growth as well as induce differentiation of cancer cells. As I mentioned before, in our lab, we've used both quercetin and chlorogenic acid, which are phenolic compounds found in dried plums, and demonstrated that we have a reduction in NF-kappa B, which is an important transcription factor that regulates a lot of inflammatory types of reactions, and a downstream effect of that was a reduction in COX-2 expression in our animals. And there was a corresponding alteration in the concentration of short-chain fatty acids in those animals as well. A prior study that was done by Yang and Gallagher a little while back showed that a powder derived from whole dried plums wound up altering fecal bile acid profiles as well as the expression of the microbial enzymes that are responsible for generating secondary bile acids. And so all of this together would suggest that we may have an improvement in colon cancer if the animals are consuming dried plums. And so for this experiment, we designed, again, a control diet that's basically an AIN standard diet, and then inserted dried plums, but as a puree. So more like what you would eat if you were eating a dried plum itself, not a dried powder. Into that diet, again, balancing for all the macronutrients. So dietary fibers, both the level and the chemical characterizations of them were identical. Protein, lipids, and carbohydrates were the same as well. So, what did we find? Well, we found that there was a significant reduction of 39% in the total number of aberrant crypt foci. Total number of aberrant crypts, like this little doublet that we've got here, went down by 42%. But importantly, those high multiplicity aberrant crypt foci that are most indicative of eventual tumor formation went down by 50%. And so here's an example of the tissue from a control diet animal that had been injected with a carcinogen, and you see that they're just littered with these high multiplicity ACF. Well, we looked at the bacteria in these animals and found some beneficial changes there as well. If you look at the animals consuming the control diet, these dark bars, and you compare what's happening in the proximal colon versus the distal colon, then we see that there is a significant reduction in the Bacteriodetes and a tendency for an elevation in the Firmicutes. Again, an undesirable profile, right? That ratio is becoming elevated again in the control diet animals that had been induced to have the disease. However, whenever we compare what happens in the plum diet, we find that we have an 84% increase in Bacteriodetes in the distal colon if they have been consuming the plum diet, a beneficial change in the distribution of that bacterial group. We also get a significant reduction in the distal colon of the Firmicutes. And you're like, well, why am I worried about proximal versus distal? It's all colon. The incidence rates for colon cancer are higher in the distal colon, especially among younger people who develop the disease. And our theory, our hypothesis going in here was that by consuming the plum diet, that we would provide a better metabolic substrate profile further down into the colon and hopefully suppress the disease process. And we do show a change in the bacterial population of some bacterial groups as a result of that. There are other bacterial phyla that don't show a change based on where they're at in the colon, but based on diet. So there's a significant increase in actinobacteria and a significant decrease in Deferobacteres in the animals consuming the plum diet. So what about biology? INOS expression. As expected in the animals that have been injected with the carcinogen, there's a significant increase in INOS expression. However, if the animals were consuming the plum diet, here in blue, you see that there's a significant reduction compared to the animals consuming the control diet. COX-2 showed a very distinct pattern of expression when you compare the proximal versus distal, with all distal animals being elevated compared to the proximal segment of the GI tract. However, again, if they were consuming the plum diet, here in blue, there was a significant reduction in COX-2 expression. The metabolomics data I'm about to show you here really quickly to wrap this up with are metabolome of the feces itself. Here we wanted to find out what was really going on in that luminal environment, characterizing the molecules that are present there. And we find that there's a significant increase in the level of sorbitol in either the proximal or the distal colon of the animals consuming the plum diet. And you may be going, what's going on with that? Well, turns out, and I didn't know that when we first started playing with them, but dried plums are actually a very strong source of sorbitol, natural sorbitol in the diet. But this has some relevance for colon cancer because it's been shown that sorbitol induces apoptosis in colon cancer cell lines and does so via a p38 MAPK signaling pathway that leads to an induction of apoptosis. In addition, we had an increase in synapate, and it does as well have some anti-cancer effects in cell lines, again through an induction of apoptosis. And finally, we have this compound 5-hydroxymethylfurfural that you cannot detect in the controlled diet animals, but is at very significantly elevated levels in those consuming the plum diet. And this compound has anti-proliferative activities. Now, all of these molecules are inherently endogenous to the plum itself. So it's making it through the GI tract, and you find it in the feces as the parent compound. But as I mentioned before, the microbiota metabolize a lot of these phenolic compounds into something else. So what are some examples there? Here I'm showing you some microbial metabolites of some compounds that are derived and have phytoestrogenic activity. Okay? Both enterodiol and enterolactone are elevated in the animals consuming the plum diet, and these are associated with protection against a variety of cancers, including colon cancer. So when you look at this whole gamish of data that I've shared with you today, what can we really conclude from it? I think it's safe to say that the bioactive compounds that we've been studying, either in their isolated form or in the food, when you combine them with an appropriate fiber source, and maybe it's the synergy of those two things together, we're able to affect the colon microbiota and their fermentation products, which winds up impacting colon health beneficially. It manages to suppress the inflammatory tone of the colon and lead to a reduction in carcinogenesis that's occurring in those tissues. Importantly, the microbiota that are resident in the colon are able to not only use the dietary fiber that's there, but they also metabolize the polyphenolic compounds that come along with that. And they wind up producing other biologically active compounds. And many of those compounds are much smaller in size and structure, making them much more able to be absorbed by us. Therefore, they influence not only the colon, but they have an opportunity to influence systemic health and physiology because they are able to get into circulation. So what's happening here? We've got the complex carbohydrates carrying these bioactives. They have an impact on the microbiota and their metabolites. We've demonstrated from our data that those metabolites are able to affect the epigenome, the transcriptome, the proteome, as well as the metabolome of the host and the microbiota. The downstream effect of that is the changes in microbiota are associated with a reduction of inflammation and cancer. Part of those may be mediated through their impact on reducing proliferation, enhancing apoptosis, reducing inflammation, and the net effect of reducing cancer. So our implications, let's go back to this original Healthy People 2020 statement. If our national goal is to avoid preventable diseases from occurring in the first place, what is it we've got to do? In order to maximize health benefits derived from foods, and for people to get that benefit, we really have to determine which sources confer these health benefits. It's not simply enough to say, eat a high fiber diet. We have to be able to tell people which fiber sources to select based on these kinds of interactions. We cannot simply generalize. So finally, I want to thank everybody who did this work. We've had a great crew of graduate students, postdocs, as well as staff in the lab that have contributed to these efforts and this data. Couldn't have done it without them. I have a wonderful group of people that I've collaborated over the years, both here at A&M as well as outside. And obviously we wouldn't have been able to do any of this if we had not gotten the funding to be able to conduct the studies. So thank you very much for your attention. Yes.
So I noticed in the obesity section of human in the serial study, it looked like there was quite a bit of variation in response to when you're looking at the metabolites or looking at the species of bacteria. So on average, it looked like there were these trends, but certain individuals had either a large response or no response. So I'm wondering if genetic diversity might play a role in that, and if so, how do you model that potential effects of genetic diversity in your animal studies?
So we have not, in the studies that I've shown you here, attempted to do anything with respect to genetic diversity in our studies. That would be a wonderful thing to try to explore. The other thing that could have contributed to the variability in the human subject study is reported compliance was good, but I don't know, right, that they ate the cereal the way that we had asked them to do so. And so that could have contributed to that variability. The other thing that could contribute to that is just what else they were eating as well, right? These people, like I said, were completely free to eat whatever they wanted to, whenever they wanted to, with the exception of consuming our cereal instead of, their Fruit Loops or whatever it was that they were eating in the morning. So if we were making measurements at discrete points in time, somebody may have been very good with their diet and kept a fairly standard diet throughout, and so the data that we have reflects what's the norm for them. Somebody else could have gone out and had, nothing all day until they got, to dinner, and then they had nothing but pizza and beer that night, So it's hard to capture human data really as reliably as you can with the animal studies, which is why I've only done one human study so far. But it's a good point about the genetic variability contributing to that. Yes?
That was a fascinating data, really, and I particularly appreciate you share a lot of published data. I was just wondering about you talking about the microbiome profile phases. Also, you're talking about the plasma metabolite profile. I know the feces is not equivalent to plasma. You
Right.
see the same thing. But do you see some sort of correlation between these two profiles? And also, which one comes first?
Yeah.
Which one is more faster rate up?
So we have done some work to verify that the metabolites, especially the phenolic metabolites that we detected in the human study, were in fact detectable in the fecal stream of those individuals. So we've done those mass spec analysis and confirmed that that was originating from the colon, or at least it was also present there. Let's put it that way. And I would have to say, for at least some of those compounds that are derived from microbial metabolism, they would have to be developed first in the colon, and that would be a much more sensitive readout than what you find in the colon. And if you look at a lot of the studies, like the work that the Taucots do, whenever people will dose individuals with a particular food source or a substrate, depending on how soluble it is and how readily digestible it is, you'll find a very rapid spike that corresponds to it being absorbed in the small intestine versus a very delayed response time showing up in the plasma if it has to get to the colon and is metabolized and released there. So it all depends on the compound as to whether it's going to show up first in the feces versus the plasma.
Last question, Clint.
So,
Sorry.
just from a broader perspective, most of the studies, and I understand when they're done that way, they're consuming plums, they're consuming this or whatever. So what happens in the context is part of the reason we don't see differences in these studies because people are consuming a mixture of things. What happens if they consume sorghum, plums, and you fill in the blank? I mean, do you still get the same thing when you have a mixture?
And you don't know until you do that work. You're never going to know the answer to that. And unfortunately, that is probably one of the things. You know, it's the entire milieu that we're being exposed to when we're out choosing the foods that we want to eat. You can wind up having some things out there that are actually counteractive to the beneficial things that you also have in your diet. They may be, in fact, fighting each other, and therefore you have no net beneficial outcome as a result of that. And that's the difficult part about doing any of these dietary studies, is trying to figure out what's going to happen in the human when they eat it. And the only thing that we've tried to do to help us be able to say that more effectively is to design our diets well so that they reflect something that would be consumed by people as opposed to these super physiological or super pharmacological doses that a lot of people use. And then use an appropriate model that will help us answer that question.
Okay, due to timing, so we will stop here. But I encourage you to talk with Dr. Turner afterwards. So let's just thank Dr. Turner for the great presentation.
I remember some of those words.
Yeah,
Barely.
it's fun to share.
Back to our unprocessed diets we go.
There we go. Or at least minimally, right? Eat the whole thing.
Yeah, definitely. So we can just set a time to discuss. Great.
Can I ask you a question? Human goes for
Sure.